Great. Welcome everybody, and we're going to tell you a little bit about the two programs, Cell and Gene Therapy and Personalised Medicine and Novel Therapies. Oh, hang on. Yeah. So these are both based at the UCL Great Ormond Street Institute of Child Health, and at the institute we run several postgraduate programs that are all listed here. So what we're talking to you about today is cell and gene therapy and personalised medicine and novel therapies that run very closely side by side, and we make each other stronger and better by doing this. So the directors are myself and Professor Patricia Ferretti, who's online, and in the next academic year, my position is going to be taken over by Dr. Conor McCann and Dr. Georgia Santilli, and we've got also programme directors for personalised medicine and novel therapies, Dr. Haiyan Zhu, who's here today, and Dr. Mark Christensen and Professor Paul Gisson, who aren't here. And we've got an administrator, Catherine Irwin, and that's her email address if you want to contact her. So the programme's got dedicated teaching associates with each module, and also teaching fellows that help with each programme. I can tell you a little bit about Mark Christensen, who's a programme director of Personalised Medicine and Novel Therapies, and is a principal teaching fellow on the Cell and Gene Therapy programme. He leads these two modules, Applied Genomics and Personalised Medicine, and he's a departmental education lead for Genetics and Genomics Medicine. He also heads UCL Genomics. So the next slide, I'll hand over to Haiyan to tell you about herself.
Hi, everyone. I'm Haiyan Zhu. I'm a director for personalized medicine and novel therapy, co-lead with Mark Christensen. I'm also module lead for novel therapy. And at the same time, also have some role at ICH and also Great Ormond Street Hospital Biomedical Research Center to lead the RNA therapy program here, and my group particularly working on RNA-based novel therapy for rare genetic disorders.
So Patricia.
Hello, I'm Patrizia Ferretti, and I am a professor of regenerative biology. So I have an interest in stem cells, and I am one of the program director for cell and gene therapy. I've been involved with this MSc program since it starts over 10 years ago. I also co-lead a couple of modules, one in stem cell and tissue repair, and the research projects. And as I've said, my interests are in stem cell tissue repair and disease modeling, and obviously cell-based therapies. I'm also involved with education as a mentor, and I am a STEM ambassador, so I'm really interested in your education.
So I'm Jenny Morgan. I'm the programme co-director currently for Cell and Gene Therapy alongside Patricia. I lead one module, molecular aspects of cell and gene therapy. So I'm interested in skeletal or voluntary muscle regeneration and the stem cells that give rise to this. Also muscular dystrophies using stem cells or other means to restore the protein that's missing in muscular dystrophies like Duchenne muscular dystrophy. And I am also interested in equality, diversity and inclusion, and we've got an Athena Swan Gold Charter here at the Institute of Child Health. So I can also introduce Dr. Conor McCann, that I don't think's here today. So he's will be the programme co-director of cell and gene therapy from the next academic year. We lead the molecular aspects of cell and gene therapy module together. Conor's research interests are in enteric nervous system stem cells and the development and repair of gut, particularly motility disorders and the physiology and pharmacology of the gut. And you can read a bit more about him on these links. Dr. Georgia Santilli will be a programme co-director of cell and gene therapy from the next academic year onward, and she leads the module clinical aspects of cell and gene therapy. She's an associate professor in infection, immunity and inflammation department, and she develops gene therapy technologies for disorders, focusing on gene editing strategies to replace or correct faulty genes in hematopoietic stem cells. Also Professor Paul Gisson is the Deputy Programme Director for Personalised Medicine and Novel Therapies, and he leads the module Novel Therapies Concept to Clinical Translation. He's a professor of paediatric metabolic medicine. He's a consultant in metabolic medicine at Great Ormond Street Hospital, and he leads the Gene Stem and Cellular Therapies theme for the Biomedical Research Centre. So he's interested in intracellular trafficking and developing gene therapies for rare disorders.
So I'm going to tell you, yes, a few words about the structure of both these MSc programs. So it can be taken as a master degrees, and that means one academic year of full studies, or you can have a sort of part-time approach or a flexible approach. So if you do a part-time, you'll have 2 years of academic study. Flexible studies can be done over 5 years. So the program involves the core modules, 5 core modules, 3 optional modules, and a research project. If you only want a postgraduate diploma, you can do that. That will be 120 credits, and the taught component will be the same as for the master degrees, but you will not be doing the project, the research project. For a postgraduate certificate, it's much shorter, and that is only a 12 weeks of full-time study, or you can do it over 2 years as a part-time student. And that involved taking the 5 core modules. So this is an example that for the cell and gene therapy, but the same apply to personalized medicine. For example, you can take 2 of the core module in year 1 and choose 2 as optional. And in year 2, you'll have the other 3 core modules, 1 of the optional and the research project. But there is a flexibility in how this is organized. And the next slide, that is just one of our beautiful cells. Can I have the next slide, please? So what is cell and gene therapy? So if you think of gene therapy, there is a very nice definition on Wikipedia. Gene therapy is the insertion, alteration, or removal of genes within an individual cells and biological tissue. And this is for the purpose of treating diseases. And gene correction through gene therapy, and obviously the modification of cells, and the two together, combined cell and gene therapy, are very important, and it's sometimes difficult to separate. So what are the stem cells? And in the next slide, you can see a little bit of a quick summary of what stem cells are, that I don't have time to go in detail now, but fundamentally are the building block of our bodies. From the embryo on, we can grow them in cultures, manipulate in different way, can be used to develop cell therapy for regenerative medicine, tissue engineering, but also very importantly, you can use human stem cells for disease modeling, and this modeling can be very important for drug screening and also for developing nanotechnological approaches to tissue repair. So why are these things so important? Why have they grown so much? It's because they have an incredible potential. And this field, as you can see from in this slide, is getting, more and more larger, with more clinical trials in different therapeutic areas, and more or less all different organs have been studied or approached for the possibility of repairing gene defects, birth defects, or for improving tissue repair. One components that you hear a lot about is the use of different vectors that are used in trials of gene therapies, and some of your project you'll be involved in the design of these vectors. So I think that's still me. Yes.
Yep,
so I'm going on what your learning outcomes are. So what we wish you to achieve by the end of your course is a basic understanding of biomedicine, have a very good grasp of statistical research methodologies that are very important for whatever you're going to do next, and also understand what are, just a symptom of different diseases. You get some example, and how this inherited disease can be treated. And in general, we wish you to acquire general skill for research science, that will be through the research project that we'll discuss a little bit more later. So the taught modules will focus on genomics, as it will be both applied and clinical component. We'll have the stem cell biology, the tissue repair and regenerative medicine. And then we have exemplify a number of approaches to gene and cell therapy and their applications. So this is an example of the structure of the course, and you can see that there are 15 credits for each of the taught modules. The module in green are the compulsory modules that you'll need to take. And the research project, as I said, only if you do the master. And then you'll have a series of choices. Some will be modules that are shared with personalized medicine. You'll be able to choose one or the other of those for the cell and gene therapy. So these are example of the work that is done here. So a lot of the work published in cell and gene therapy has been really as a forefront, particularly in pediatrics, because we are associated with the pediatric hospital. And you'll be able to look, if you're interested, some of this paper, and there have been also some very recent exciting news about the child being cured through cell and gene therapy by one of our colleagues, Professor Qasim, using a CAR-T therapy approach.
Over to Haiyan.
Hey, hi everyone. Again, I'm Haiyan Zhou. I'm going to give a very brief introduction about our program, which is personalized medicine and novel therapy. So this is our program team, and this. No, don't worry, Jenny. Carry on. This program is really about the next generation medicine, and healthcare is applicable to clinicians and healthcare workers, and also people working in industry and pharma, and also related area. Jenny, next.
Yeah.
Yeah. So currently our medicine is the system is one treatment fit all of us. Doesn't matter what you have, as long as your diagnosis is that, and that's your treatment. Does it work? No, not really. In most people, yes, work, but we do see that for some patients it doesn't work, and some you will also see the adverse effect. So to tackle this in the future, the future direction is we want more personalized treatment. So to make this, we have the one treatment. For patients, we will check a biomarker, and we will check genetics, and to understand what's different among patients. And then we can change our treatment to make it more personalized. So that's our aim, to enable the right drug at the right dose to the right people and at the right time. To make it, we need to involve lots of things. For example, we need to involve the new apps and solutions. We need to involve multi-omics for biomarker studies. We need to involve the new computational biology tools. And also we need the new therapeutic strategy, new pharmacogenetics. And we need also with the support from patient advocacy group and new ethics and the social science and clinical trial regulation and new trial design. Also, we need to some other cool thing, for example, the wearable and medical device. If you have read the BBC News, you were here yesterday, that's something very exciting. They talk about the advanced science, you know, something. Have you watched Avatar during Christmas? So now scientists use the same technology and in clinical trial design. So patient can wear this motion, the device. It's like, and then capture it's called the motion capture. That device can use to indicate if the treatment work or not. Even it's a very small change, that device can pick it up. So that's the future of our medicine will go to. So back to personalized medicine and novel therapy, the aim of our program is for you to understand the basic scientific principle underlying the major application of personalized medicine, novel therapy, computational science, and also understand the regulatory issues behind to support the clinical trial design and other ethic issues, and also biomarkers and all this AI machine learning in medical settings. And we also have, like Patricia has introduced, we have research project as well as the same requirement. Next. And this is the list of our publications. Of course, we are more in the last few years. There are lots of new things have coming out. Yeah. This is about our modules. The same, we have 6 compulsory modules, including personalized medicine, novel therapy, computational biology, applied genomics, and applied statistics. So this is a 5 taught module plus a research project. And also students will have the chance to choose some module from the optional module, which highlight in blue. So something we shared with cell gene therapy.
Oh, hang on, did I go too far? Over to
yes, that's what I need. So how will you learn? As we have said, a lot of the people will be teaching you will be the people who have published and work in these areas that are really the forefront. So will be either the boss themselves or people from their laboratory who have done the work. So that's very exciting. And now the learning is mainly face-to-face, so will be in person and in the classroom, and you are expected to attend. There will be also some synchronous online work, and obviously we hope not to have to do too much of that, so no pandemic will affect our teaching next year. The assessment will be either online, there will be a number online, but there will be in person, giving, for example, presentation, and that will depend on the structure of the module. And the vivas at the end of the year in September, after you submitted your project, will be in person. So this is just to summarize all the different ways. So what we hope to give you on both programs is really a varied approach to your way of learning and your way of taking exam to really equip you in different areas, you know, just to get you ready to the real world, whether you decide to go on and do research, or if you want to work in an industrial setting, in publishing, in medical regulatory bodies, whatever. And so you can have, just a really lecture, seminar, or a presentation. We'll have access to all the facilities that there are at UCL. I think that summarizes that. The research project, a bit more detail of the research project. That is really, very important in both modules. And this is largely done in laboratory projects, but there are also bioinformatic projects that are particularly relevant to personalized medicine, but to both projects. But really, we want you, whichever type of project you choose, and we provide a portfolio of projects. for you to choose from
Sorry.
it's all right, is to use a variety of methodology to try to answer questions that we expect you, together with your supervisor, to set very clear, and can be either in cell therapy or personalized medicine. So really, we want you to learn to formulate questions and develop approaches to address them. We don't expect you to do a huge piece of work because the time is limited, but to really become confident with all the tools, become really adept to exercise a critical approach, both to the literature and to your own research. So if we summarize the learning outcomes, really want ability to formulate a research question, get a good grasp of the research background, the method, understand the design, and really to design, including control and good experimental structure, is essential to get good answer. So you want them to be able to analyze data and develop the appropriate tools for that type of analysis. And we want you to be able to present your research, both orally and in writing, in an effective way. So these are examples of projects that we have offered, and we have, a range of between 40 and 50 projects, depending on the years, and the university college is huge. So if we don't have project within our institute that is running the program, we have a lot of colleagues within this college. And so we usually manage to match students who have a particular interest in an area with an appropriate supervisor. So for example, for cell and gene therapy, we have in silico investigation of therapeutic potential and impact of trophy regulation in Duchenne muscular dystrophy, or analysis of Netherton syndrome, that is a rare skin disease, application of antisense oligonucleotide therapy in neurodegenerative diseases, and repositioning of a drug for rare diseases, novel cancer therapy. And example of projects that have been taken by students, personalized medicine, are, for example, investigating patient pharmacogenetics profiles using DNA microarrays, or using single nucleus RNA sequencing data from postmortem brain tissue. And that was a project aimed at better understanding Alzheimer's disease. You can look at epigenetic biomarker, neurodegenerative diseases, or investigate autophagy, for example, in metabolic bone diseases. And here there is an example of how you'll be busy, how your timetable will look. So you are a step above undergraduate, so you have to gear up for that. And you'll be doing a number of modules. So in this case, in blue, in green are the compulsory modules for cell and gene therapy, and in blue are some of the optional ones that you can have, for example, in the first term. And if we go to the next slide, we have a summary that is a mirror of what are the green compulsory module for personalized medicine, and in blue you can see the optional one. And obviously you'll have statistics that is very important for both programs that will be run in parallel. And the second term you'll have most of the what we call the intensive modules. Term 3 is all dedicated to the project.
Okay, so I think over to me then. So yes. These are the modules on our two programs, and I'll go briefly through each of them. There are quite a few. So the first is critical thinking and scientific communication, and in this you learn to critically analyze papers, how to present your work in writing and then orally. And this is what you have to do: prepare a lay summary, give a 3-minute talk, and this will improve your written English for research. Oh, I think I've done 2. So The next one is clinical aspects of cell and gene therapy. And in this, you will be familiar with gene therapy strategies that are either in or about to enter the clinic, and understand the processes, principles and regulation of bringing cell or gene therapies from the bench to the bedside. And during this module, you'll have the unique opportunity to learn from people that are really pioneers in the field of cell and gene therapy, from different scientific backgrounds and expertise, and this will help you choose your future career path. In applied genomics, you'll design an experimental genomic pipeline and understand the importance of sample quality, next generation sequencing, single cell technologies. You'll have a case study to look at. You'll analyze and interpret the results, and you'll discuss and debate ethical considerations of using genomics in research. And at the end, you'll understand how genomics can be applied to many different fields in science and medicine. And this is a really interactive and exciting module. In molecular aspects of cell and gene therapy, you'll really be led to understand in real depth about how complex it is to treat genetic diseases, and be familiar with the molecular biology involved in cell and gene therapy. So it's a really in-depth understanding of how things work. And you'll learn state-of-the-art approaches, including novel editing techniques such as base editing, and you'll also have a perspective from somebody in industry, from a gene therapy startup company. In stem cells and tissue repair, you'll learn about stem cell biology, tissue repair, the potential of using stem cells for modeling human diseases and for clinical applications. You will understand how important multidisciplinary approaches are. Again, regulatory and ethical issues in the stem cell field will be discussed, and you'll have a selected topic that you'll present in writing and in orally. So again, in this module, you'll be taught by people who are at the forefront of their fields and do projects in their labs. And you may also have the opportunity, you will have the opportunity to attend research seminars, and also hopefully the one-day London Stem Cell Symposium. So applied statistics in health research, I think everybody has to do this, and you'll learn how to interpret the statistics presented in published work, and about areas to consider when conducting your own research. So this is taught in a newer and exciting way with interactive polls, lots of hands-on practical exercises, and plenty of opportunity to ask questions. So you can study it all online, where the content's broken into short videos and presentations with quizzes for each chapter. So in clinical genomics, genetics and rare diseases, you'll learn about methods used to screen for genetic disorders in the NHS setting and in research, including prenatal diagnostics, ethical implications, again, gene-based therapies gene-based therapies used to treat these disorders. So you'll understand the really recent advances in molecular medicine and genetics and how this impacts on work in the clinic. In molecular biology of normal development and birth defects, you'll understand how we human embryonic development, and how the mechanisms of birth defects and what they cause, how they can be diagnosed and prognosis of birth defects, and the development of novel therapeutic or preventative strategies. In molecular and clinical aspects of childhood cancers, this aims to provide insights into the etiology of most common pediatric cancers and the molecular and cellular basis for these. And also you'll understand how treatment has improved outcomes, recent new treatment, and how novel technologies coupled with new understandings lead to improved diagnosis and therapeutic interventions. So pharmacometrics, gosh, I can't see it. So say it, you'll learn how to fit pharmacokinetic, pharmacodynamic models to clinical and preclinical data. And it's an area, this particular area is an area of skills shortage in the industry, but it increasingly underpins modern informed drug development. So it's a really exciting module, and it could lead to career opportunities. Computational biology for personalized medicine. You'll learn how bioinformatics and genomics have shaped new approaches to personalized medicine and state-of-the-art experimental and computational technologies applied to this. So you'll learn about current bioinformatics and the challenges in genomic medicine and how to use computational biology as a tool for personalized medicine. In novel therapies from concept to clinical translation, and this will teach you about the clinical translation of novel therapies all the way from lab work at the bench to the bedside. And it's got lots of module content that's listed here. So personalized medicine, again, you'll learn lots about 21st century healthcare, biological and digital markers, the use of medical device technology that I think Hyan touched upon, the importance of data sharing, stratifying patients to get the right treatment to the right patients, and virtual reality and machine learning, and how personalized medicine is becoming more and more a part of scientific research. And this we've done through case studies. You'll also consider ethical considerations and also some skills that will allow you to be an entrepreneur in the field. So in cell-based immunotherapy, you'll have a critical understanding at the end of it of CAR T cell-based immunotherapies for cancer. And you'll understand how the immune system, how you can engineer it, how to design and manufacture CAR T cells, and preclinical and clinical applications of these immunotherapies, and ways to overcome current limitations. I think, is this the end? Then I pass over to Hyan, I think, do I, Hyan? I can't remember how far I've got here.
Yes, Jenny, I can pick
Thank
up.
you.
So this is all about the assessment types for all the module we have mentioned. We have a wide range of assessment type that include, you can write, you will give time to write an essay, give a topic, and you will also have this question written examination, for example, multi-answer questions, and you will also have the chance to prepare a poster, leaflet, to do a case study, or and some module will also ask you to give oral presentation. So it's a definitely wide range of assessment. Yeah, and this is some key dates keep in mind. And if you start the both program, and one of them, or not both, so the induction will happen at the end of September this year. And then we will give the module presentation the same similar time, end of September, early October. So that's the one. The top one is at faculty level and followed by program level. You will also be given the library session training. That's usually the first session to start with. That's in early October, and then after that, you start select your module from your program, and the module lead and also the module lead, the program lead will help you with that and also our admin team. So that one you need to finish before 6th of October, and then the first module start 9th of October this year. Study at UCL is wonderful. You will receive support available all year around. So that's here we list that's for academic research, academic writing. You will have those, and also the reference. You will learn EndNote, library training. You will learn how to search literature, and also you will have the training of tutorial. You will be offered personal tutor, so you will be linked. Each of you will be linked your own tutor, and also you will have the revision session, exam preparation, and so the module and program will support you. And also for some programs, for example, cell gene therapy, you will also have the chance to apply for scholarship. And here is a website. Any other questions, you can use this website, Ask UCL. And apart from them, the other support, for example, during the pandemic, we have this support. And then for well-being, we have a long list of mental health and well-being, healthcare, disability support, and also other things related to your day-to-day life, for example, travel and student uni, student loan, and advice on remote learning. All these issues, yeah, you can find from Ask UCL. The activities at UCL, and not just the scientific, but we also support you to develop other skills. For example, I think Patricia or Jenny already mentioned the 3-minute thesis. It's very popular usually at the beginning of August, once most of you finish your research project, and that's the time you showcase your study. And students usually are encouraged to join journal clubs from your own research team, and also you can organize your own journal club, and the department seminars and conference. Library skill, of course, it's essential, and writing skill, all this training, you will receive the training and to develop this skill. So where our student, this is about employability. Our program aims to equip students for career in research, education, medicine, and so you have lots of choice. You can you can go to after our program. You have a chance to go to university as a researchers and also in industry. You could also take role involved in clinical trial as coordinator, or maybe as a clinician or scientist, and also allied health professional. And we also have some students, brilliant students, they also set up their own company. Every year we have 1 or 2 students, our staff, that have their own company. And also some students work in the law and legal. And of course, quite a lot of our students also have successfully applied PhD from UCL, Oxford, and many universities in the UK.
So
question time.
Question time. I'm going to stop sharing. And then do we stop recording and then
I'll stop recording?
Yeah. So